Uday. I am currently pursuing MSc Microbiology. In this presentation, I am going to talk about bioremediation with the special emphasis on the advanced bioremediation techniques. Before entering into the presentation, I would like to pose a question. Can we reprogram nature to undo the damage that we have already done? The answer to this question is not an arbitrary yes or no. The answer lies in a complex field in between. Through this presentation, we are going to explore that complexity and enlighten ourselves with a meaningful answer at the end. Before entering into the actual bioremediation process, I would like to discuss about the impact of pollutants in a broader sense. Globally, over 400 million tons of plastic waste are produced annually. Air pollution nearly contributes to 9 million premature deaths per year, and nearly 8 trillion US dollars is being invested to address this particular thing. This obviously has a huge amount of economic and social impact in terms of food security, social inequality, migration, and associated aspects. This particular graph essentially talks about the need of remediation. The y-axis represents the environmental impact index and the x-axis represents the time units in terms of decades. If we look at this particular curve represents the condition where pollution rate is more than that of the self-cleansing ability of the environment, which is nothing but remediation. This particular curve represents the condition where pollution rate is less compared to that of the self-cleansing or remediation ability of the environment. We can see a huge difference in terms of their performance ability, which talks in favor of the need of remediation in order to achieve the intended intention. Nature intrinsically has the ability to restore the environmental balance. Every ecosystem possesses self-cleansing ability and mechanism that acts as a natural purifiers. Some of the notable natural self-cleansing mechanisms are dilution and dispersion, sedimentation, microbial degradation, biogeochemical cycling, and volatilization. Dilution and dispersion is very straightforward, where the concentration of a particular pollutant is diluted via rivers, streams, and atmosphere. Sedimentation and adsorption is the physical method where pollutants are sedimented and adsorbed on the physical materials so that bioavailability of those materials is less. Microbial degradation also is very straightforward, where particular microorganism has the ability to degrade particular pollutants. So employing those microorganisms to achieve the intended intention is the environment's way of achieving the bioremediation. Biogeochemical cycling is naturally happening where carbon, nitrogen, sulfur are constantly being transferred from one form to another form in the ecosystem. Volatilization and photodegradation is utilization of these particular pollutants by plants as their for the production of their metabolites. While these natural processes offer a very huge amount of advantages, they also carry a limitation with themselves. This particular graph illustrates the fact that a particular natural self-cleansing mechanism need not be efficient in achieving the intended intention of pollutant clearance. If we consider natural cleansing efficiency in terms of percentage in the y-axis and particular pollution type or load in the x-axis for different pollutants like organic waste, nutrients, synthetic pollutants, heavy metals, and other things, this particular line depicts the particular natural cleansing efficiency below which intervention is an absolute necessity. If we consider organic waste and nutrients, they are efficiently cleansed via natural cleansing mechanism and need not have to be dependent on the human intervention or any sort of intervention from outside. But whereas synthetic pollutants, heavy metals, and other things which are predominantly created by human, it requires human intervention for their efficient maintenance and cleansing. Environmental remediation in general can be categorized into three categories: physical remediation, chemical remediation, and bioremediation. The name are pretty straightforward. Physical remediation is the physical removal of the pollutant. Chemical remediation is the utilization of chemicals or chemical processes for achieving the remediation process. And bioremediation is the utilization of biological agents or their producers in order to degrade, detoxify, or transform the environmental pollutants into a less harmful product. In this particular presentation, we are going to discuss more about the bioremediation process. Compared to physical remediation and chemical remediation, bioremediation is more eco-friendly, cost-effective, it is scalable and adaptive, it is effective against resistant pollutants, sustainable and self-regulating, which gives it an extra edge compared to physical and chemical remediation. Some of the classical bioremediation techniques are bioaugmentation, biostimulation, phytoremediation, composting, and bioventing. Bioaugmentation is fairly simple. It is the introduction of specific pollutant degrading microbial strains into the contaminated environments. Biostimulation is the stimulation of the indigenous microorganism present in the particular environment to provoke them to degrade the particular pollutant present in that particular environment. Phytoremediation is the utilization of a particular pollutant by a particular plant to produce their metabolite. Composting is the aerobic digestion or biodegradation of organic pollutants in soil by microbial consortia. Bioventing is like extension of biostimulation where oxygen is supplied to the contaminated soil in order to revoke the aerobic microorganism that has the ability to degrade the particular pollutants. All these techniques offer their own set of merits and demerits, and depending on our intention, we are going to specify the particular type. As I previously mentioned, though the classical bioremediation techniques offer a very fruitful result, they also carry the burden of limitations. Like they offer a limited pollutant range since we are specifically using specific microorganism to degrade specific pollutants. They are highly dependent on the environmental conditions. The degradation rates can be slow or dependent on the environmental conditions. The bioavailability of pollutants is a huge risk while dealing with the bioremediation. Competition in survival issues, since we are utilizing biological agents, competition is a natural phenomenon that can occur. Monitoring and controlling this bioremediation process can be challenging compared to physical and chemical remediation. This particular graph shows the comparative effectiveness of classical bioremediation against the advanced bioremediation or other thing. If we consider remediation efficiency in terms of percentage in the y-axis and environmental complexity or pollutant type in the x-axis, this particular curve represents the controlled laboratory condition. If we look at this particular curve, we can see that as the complexity of the pollutant increases, the efficiency remains more or less same. But if we consider the field deployed conditions or real world condition, as the complexity of the pollutant increases, the remediation efficiency decreases. This gap has to be filled, and this demands for a critical need of innovation which can be addressed via advanced bioremediation techniques. Advanced bioremediation techniques need not have to be a replacement of classical bioremediation techniques. Rather than advanced bioremediation techniques can be extensions of classical bioremediation techniques, which offers either specificity or broader spectrum depending on the intended intention that we are striving to achieve. It involves the deliberate enhancement of the natural biological system using modern scientific tools like genetic engineering, microbial community engineering, nanobiotechnology, or electrochemical stimulation to accelerate the pollutant degradation, expand the substrate specificity, improve the process control, and enabling them to thrive under stressful conditions. For simplicity, These approaches can be broadly classified into four categories, which are genetic engineering based bioremediation, consortia based bioremediation, nanobioremediation, and electrochemical based bioremediation. In this particular presentation, I am going to only discuss about the genetic engineering based bioremediation and consortia based bioremediation. Genetic engineering based bioremediation. The name is pretty much straightforward. Genetic engineering based bioremediation incorporates genetic modification of the microorganism in order to achieve the improved degradation efficiency, expanded substrate specificity or substrate range, enhanced resistance to environmental stress, and precision in terms of accelerated pollutant removal. There are several strategies to achieve this genetic engineering based bioremediation techniques. Few of them has been listed in this tabular column. Pathway engineering, plasmid mediated gene transfer, stress resistant engineering, synthetic biology, and reporter gene systems are some of the notable strategies. Pathway engineering. In this particular strategy, we know that particular microorganism has the ability to degrade a particular pollutant because they have a specific pathway to do so. Those pathways are guided by a set of genes. If those genes can be engineered into another microorganism with suitable environmental adaptability, we can achieve the intended intention. Like for example, XYIE is a particular pathway which is responsible for degrading aromatic hydrocarbons like toluene and xylene efficiently. Pseudomonas putida can be engineered with this particular pathway or the genes responsible for this particular pathway in order to achieve this degradation of aromatic hydrocarbons. Plasmid mediated gene transfer is also very similar to that of the pathway engineering, but instead of transferring genes to chromosomal level, here we are utilizing extra chromosomal genetic material for transferring of particular genes, which gives it extra work for discussed improved degradation efficiency and expanded substrate range. Like for example, MAR-A or MAR-B gene can be incorporated into plasmid and transferred to that of the E. coli, in which can convert toxic mercury Hg2 plus ions into a less toxic Hg0. Stress resistant engineering. Particular microorganism has the ability to tolerate stress while others do not have the ability to tolerate stress. So genetically modifying those microorganisms while equipping them to tolerate stress can be a good way or good strategy to achieve the intended bioremediation. Synthetic biology, where we construct the whole genome profile and genetic circuit to achieve the intended intention of pollutant degradation. Reporter gene systems work more like a sensing tool than that of the degradation technique, where the system can efficiently sense the pollutant present in the environment rather than degrading them. This is the overview of whatever I discussed till now. And the advantages this particular genetic engineering based bioremediation offers over classical methods is that it offers a broader pollutant range, it enhances degradation speed and environmental tolerance, it is customizable in terms of metabolic pathways and specific pollutants that it is going to target. The possibility of self-regulating is higher compared to natural things, but it also poses a set of challenges or ethical considerations. Since we are using genetic engineering as a tool here, the rules are pretty much stringent and regulatory and biosafety constraints are very huge. There is always a risk of gene loss into the nature which can be hazardous. The second technique is consortia based bioremediation techniques. Consortia is nothing but synergistic community of microorganisms which works in a coordinated manner in order to survive under whatever condition they are present. The collective degradation of complex pollutants more efficiently than the individual strains is the key idea of utilizing consortia based bioremediation. This tabular column addresses the limitation of utilization of single strain for bioremediation and how It is advantageous for utilizing consortia-based bioremediation to address those limitations. While utilization of single strain can limit the metabolic range, consortia-based remediation can be work in a complementary manner to achieve the same effect. Single strain can be sensitive to a particular condition, but consortia-based system offers a complementary approach where if one particular microorganism is sensitive to a particular pollutant or a condition, the other will take care of the intention. Incomplete degradation in case of single strain bioremediation can produce intermediates that can be very harmful, but in case of consortia based bioremediation, the system is designed in such a way that intermediates are also utilized for the production of less toxic pollutants, less toxic products. These are some of the mechanisms utilized in consortia-based bioremediation like metabolic complementation, cross-feeding syntrophy, quorum sensing and communication, and biofilm formation. Metabolic complementation is nothing but utilization of a particular metabolite produced by a particular microorganism in a system by other microorganism in the same system, so that they work in a coordinated manner. Like the consortia of Pseudomonas and Bacillus targets crude oil hydrocarbons very effectively than that of individually. Cross-feeding syntrophy is nothing but exchange of metabolites, cofactors or electrons among the species of the system. Like aerobic and anaerobic bacterial consortia is very efficient in tackling chlorinated solvents. Since it is a synergistic system, there will be intracellular communication, and this communication efficiently guides towards degrading a particular pollutant with reduced amount of intermediate produced. Like a consortia of fungus and bacillus can particularly target synthetic textile dyes, which can be very harmful. They can also form biofilm, which can be extension of intracellular signaling that I previously mentioned. Formation of biofilm can additionally they offer the advantages of broader substrate range, greater environmental resilience, stable operation, and reduced accumulation of toxic materials. But they also poses limitations like it is difficult to control interspecies balance. There will be competition between the members of the species in the particular synergistic consortia, risk of metabolic inhibition among the members. Syntrophy is not always the point of achievement. This particular graph represents the comparative remediation efficiency of classical methods, genetic engineering based approaches, and consortia based methods. The y-axis represents the estimated remediation efficiency and x-axis represents the pollutant type of different pollutants. If you consider hydrocarbons, consortia based bioremediation is very efficient in mitigating it, while chlorinated solvents and heavy metals can efficiently mitigate it via genetic engineering based approaches. Up until now, we have discussed the advanced bioremediation techniques in the laboratory scale. But in order to emphasis on the significance of this particular technique, I am going to present this particular case of arsenic bioremediation in South Asia, predominantly India and Bangladesh. Groundwater contamination affected approximately 140 million people in Ganga Brahmaputra basin. Chronic exposure to arsenic poses a severe public health risk for people of both the nations. Instead of utilizing chemical precipitation or adsorption, scientists decided to employ consortia-based bioremediation. They used indigenous microbial consortia of Pseudomonas, Bacillus, and Geobacter and algae, which performed arsenate oxidation, arsenate reduction, biomineralization, and bioabsorption. This particular graph represents arsenic consortia before and after remediation. This particular gray color bar represent the baseline arsenic present before remediation, and green bar represent the baseline remediation after remediation. They considered 4 pilot scale regions, which are West Bengal, Bangladesh, Bihar, and Assam, and we can see that there is a significant decrease in the arsenic concentration after the remediation process, consortia based remediation process. This also represents the same thing, but along with the implementation cost index, which is indicative via this particular scale. This was a huge case and had a huge geopolitical and policy impact. This was the first bioremediation driven diplomatic collaboration between the two nations. WHO and UN recorded this as the largest mass poisoning in history, and the policy integration of microbial innovation becomes much more stringent and applicable after this. Finally, for the question that I asked earlier, whether we can reprogram nature to achieve the intended intention, the answer is yes, but we have to recalibrate it efficiently in order to reduce the harmful effects. Advanced bioremediation proves microbial systems outperform conventional methods. Consortia based and genetically engineered microbes offers a very synergistic and targeted pollutant degradation. Some of the case studies talks about its influence in the real life also. Building global microbial banks and policy science collaborations will strengthen this disaster response. These are some of the references. Thank you.